consultation. I'll mind it. I'll mind the waiting room. Okay.
I'll try to behave. Thank you very much, Tony. It's a pleasure as always to be here with you guys, and we are back at it. So it's this introduction to sponges, which is actually an introduction. If you guys have never attended any of the in-person or online workshops with me, or it's more to refresh your memories about sponges. I start first of all, tell you who I am if you don't know myself, even though I'm pleased to see a few people I know in the audience. So my name is Maria Vittoria Marra. I work as Education and Public Engagement Officer at Galway Atlantaqauria. And you may wonder, so why do we talk about sponges? Well, sponges are actually the original reason I'm in Ireland. You can tell from clearly from my name and my accent, I'm not Irish, I'm Italian. I come from this lovely part of the world that is the Strait of Messina, the geographical center of the Mediterranean, where I've become a marine biologist and ecologist. During my master's studies, I moved to the north of Italy in a place called Genoa, where I started studying sponges, and I focused on learning how to farm them underwater to produce biomass to be used to extract compounds of pharmacological interest. I will tell more about that later. And that was my connection to Ireland, because I got very passionate on the topic, and in Galway, in the University of Galway and UIG at the time, in 2013, had the opportunity to start a PhD focused on understanding the biological mechanisms underlying the bioactivity of a number of sponge species. You will get to know more about later on during the presentation. But so let's start talking about sponges now. Now that I work in the aquarium, I'm actually pleased to see the fact that a lot of kids actually know that sponges are animals, but many adults still think that sponges are plants. And if you look at the pictures on this slide, it might not be even that surprising that they are confused because of how they can look sometimes, but sponges are actually animals. They are our ancestors. Basically, there was for a certain time confusion in decisions about the idea, are the sponges actually our ancestors or the gum jellies? That was defined a few years ago when a paper cited on the slide there proved with molecular data that the sponges are the sister group of all the other animals. So, but they are, in fairness, pretty obviously quite simple organisms. They are, I always describe them to the kids like essentially a bunch of cells kept together by some sort of matrix that is made mainly of collagen, and this whole mix of cells and collagen creates this body that is how can I say in English? It's pierced by a very high number of tiny pores. In fact, the scientific name for sponges is porifera, that means pore bearers. So through all these tiny little pores, the water gets inside the body of the sponge, and all the food that the sponges can extract from the water is filtered through their body, and the water is expelled through larger holes that are called oscules. So this is the simplest explanation of how a sponge works. In fairness, they are much more complex than that. It has been counted that they have over 30 different types of cells, but still, probably in terms of sponge cells, the main character are these guys that you can see in the bottom right corner of the picture there. They are called choanocytes. They are pretty interesting cells. They have this long flagellum that is like a wipe that creates, basically the movement of this flagellum creates all the current of water inside the body of the sponge. So it basically sucks the water into the sponge through the pores and out through the oscule. And as you can see around this flagellum, they have this sort of skirt almost that is made of all this extension of the cell that are called microvilli. And these are, this microvilli creates a sieve around the flagellum that is where the microbes or the detritus, the organic matter that the sponge feed on is captured. These are a few pictures of probably these are actually this is a little too complex, but these are pictures taken at the electron microscope of these cells, where you can see very well this skirt of microvilli. And if you look at the gray picture on the top right corner, so you see the circle of section of this microvilli, and the circle at the center is the flagellum. So the way these choanocytes are organized along inside the body of the sponge, they can be along all the canals that connect the pores to each other, or in specific areas of the sponge body, is one of the way sponges are classified. There are other ways related to their skeleton, but that we will see in details later on. But anyway, sponges are classified in 4 different main groups or classes. There are the Calcarea, that are the ones with a skeleton made of little skeletal elements called spicules that you will get to know better later on during the workshop. And in this group, they are made of, they have calcareous nature. Then the demosponge, that is the largest group of sponges. Around 90% of species belong to this class. Then the Exactinellida, that, as well as the demosponge, have spicules, so skeletal elements made of silica. And then the Homoscleromorpha, that is the newest class, so the newest group of sponge to be classified, and it's very, very small. It counts only around 100 species. So sponges can be found in many, many different type of environments. In the ocean, obviously that's why we're here today, but also actually in fresh waters. In fact, the first picture on the left-hand side with the green sponges, those are examples of sponges that can be found in rivers and lakes. As regards the ocean, they can be found in sheltered areas, in caves, but also in very exposed areas. They can be found in cold water, even in polar waters, or in temperate water, in tropical waters, basically in every type of climate. And also they can be found in shallow waters and in very deep water as well. Like this picture here represents is a picture of a sponge garden that was found in the deep sea. The deep sea in Ireland is an example of environments like this can be found, and these are of great importance from a biotechnological perspective, as I'm going to explain later on to you today. And I forgot, actually, I skipped this slide because when I said that sponges can be found in tropical environments as well, I forgot to mention that actually some scientists believe that sponges can actually replace coral in tropical environments if, as you know, as we know, corals are affected because of climate change by bleaching, which is something that anyway scientists are trying to find solutions for. But it could be possible that sponges may replace corals as the main ecosystem builder in these environments with the effects of climate change. But so that would be the reason why sponges could become a key element in this sort of the environment is because they play very important ecological roles already. So they, first of all, I mentioned before the way they feed themselves. So we can define them as filter feeders. Because of that, they tend to transfer nutrients from the water column to the bottom of the ocean. So they act like benthic pelagic coupling actors. Themselves, they represent a source of food for many other animals. Nudibranchs, starfish, sea urchins, fish are some examples. And because of the fact that they filter water, they can help control eutrophication phenomena, so reducing the amount of nutrients in the ocean that would trigger the development of algae. They, in the case of the sponges, which skeletons that is made of silica, they can represent a very important storage site for this important nutrient, very important nutrient in the marine environment. If you think that some microalgae like diatoms, their the case containing their cells is made of silica, and also they actually represent a source of shelter for many other organisms. Sometimes these sponges develop symbiosis with these organisms. These are some examples in the pictures here. You may be familiar with some crustaceans, crabs, living, carrying sponges on their shells. The reason why they do that is for camouflage, but there is also another very important reasons, and that is I mentioned earlier on. Sponges produce compounds of pharmacological with pharmacological properties, as I said, because these compounds are actually toxic. They can be used as they are used by sponges themselves as chemical weapons, because sponges are sessile organisms. They live mostly attached to the substratum. They don't have any way to defend themselves because they don't have teeth, they don't have claws, they don't have spines. What they have is the possibility to produce these toxic compounds to keep away competitors and predators. So potential symbiotic organisms like crabs or brittle stars or worms or others can actually take advantage of these toxic compounds produced by the sponges to protect themselves. But sponges establish symbiotic relationships also with organisms we can't see with our bare eyes. So with many different types of microbial organisms, so bacteria, viruses, many different types of fungi, microalgae, they have all been found in sponges. Sometimes sponges host so many microbes within themselves that these microbes represent up to 35% of their body mass, which is huge if you consider that ourselves host only in between 1 to 3% of microbes in our body. And this is same concept as we were saying before. Sponges offer a lot of benefits to these microbes, but also they benefit of these microbes themselves, because these microbes produce their own array of bioactive compounds. So sponges can further benefit to protect themselves from the compounds produced by these microbes. So it's for the sponges is not exactly the sponges that we find in the natural environment. The real sponges is not exactly as the case of the sponges we use to wash our plates, as you can see on the comic here. So the presence of this massive amount of microbes in sponges can be very, very useful for themselves, but also for ourselves, because this broad variety of compounds, both produced by the sponges and the microbes they host, as I mentioned before, can be exploited by us, because the scientists have found these compounds to have pharmacological application. Unfortunately, I see this. I have two pictures that sometimes they work, sometimes they don't on this slide, but I had two examples of sponges there from which compounds with anti-tumor property that are now on the market as like official drugs have been extracted. So which is massive to think that we can treat diseases like cancer thanks to the sponges, but not just that. The compounds extracted from these animals have been found to have antimicrobial, antiviral, anti-malarial, anti-psoriatic, anti-inflammation, and many other applications. So it's so important, this role that sponges can have for our own health, that they have, in the last few decades, that have been a very important subject of study because of the extraction of these compounds from the shallow waters to the deep sea. Now, currently, there are many scientists focusing on the compounds that can be extracted from sponges from the deep sea, and because very high amount of compounds, sponge biomass is necessary to extract these compounds. Also, it's very, very important to be able to produce these biomass, so to farm sponges. And for sponges can be farmed in situ, so it means in the environment, in the natural environment, in the ocean mainly, or ex situ, so it means in aquariums, so in tanks or other types of aquaculture facilities. There are scientists trying to develop pure cell cultures, and also this weird word that you see there, primorphs, is another frontier of production of these bioactive compounds. Primorphs are essentially aggregates of sponge cells that can be cultured in lab conditions much more easily than cells. So this is just pictures from my own master thesis. I was farming this sponge called crambe crambe, but this is actually nothing new at all. Sponges have been farmed for centuries, because sponges have actually been used for centuries. We are all probably familiar with the use of sponges to wash ourselves, because their skeleton is very soft, but also very resistant at the same time. So it's a great tool for that, but also can be applied in different ways, like for instance as a painting tool. And because of all these traditional, let's say, uses of sponges, the the farming of these animals dates back to at least the 18th century. So this was just a very brief overview of some aspects of sponge biology, ecology, and the uses of the sponges. I'll start saying this, and I will repeat it at the end of the workshop today, that we, with the guys of Research Ireland and with our other collaborators in the zoology department of the University of Galway, we would like to organize an in-person sponge workshop in the aquarium here soon enough, if we get an interest from you guys and other divers and snorkelers around the island. So we will be able to talk in much more detail about the many different aspects of sponge biology and ecology. But now I want to talk about something that is strictly connected with the reason we're here today, that is the Sponge Project. So the Sponge Project is one of the projects run by Research Ireland, one of the citizen science project that is focused on trying to increase the number of recordings of sponges in Ireland, as well as the knowledge of these very fascinating, sorry, I'm biased, animals among divers and snorkelers, and hopefully beyond that. So the Sponge Project, what's the idea behind it, is for you guys, when you are out in your natural environment, that is the place where you dive or snorkel, or just go rock pooling, if the tide is very, very low and allows that, is to if you are interested in a part, if you see a sponge that you would like to know what that sponge is, and hopefully with that being able to recognize it and identify it in other circumstances, you can collect a sample and send it to us. Our collaborator in the University of Galway will carry on the identification of the species for you. And now what I want to tell you for the rest of the workshop today is how to collect these samples, how these samples are identified, and what else you can do if you want to join the Research Ireland Sponge Project. So first of all, I want to talk about the fact that sponges are difficult to identify compared to, well, first of all, I'm not a taxonomist, properly trained. No, again, I was properly trained, but I'm not, I can't define myself as a taxonomist. That's not what my research was about. But, and I'm not aware if for all the different groups of animals is as difficult, but definitely sponges are not exactly the most straightforward animals to identify, not just because there are more than 8,000 different species. Also, the same species can look pretty different in different circumstances, because these animals are great to adapt to the environment where they live, but also they can change their own body shape or growth form based on characteristics related to themselves or their life cycle or their health status and things like that. So the plasticity is definitely a big characteristic of these animals. So I have a first example here is one of the species I've studied during my PhD in the University of Galway, that is called Aliclona similans, is underwater in sheltered condition. It grows as branching sponges. It can really get big, creating big sort of fans, but the same species, if you look at the picture on the right-hand side, in intertidal conditions, in very exposed areas, grows almost encrusting. So you see that this is already an example of how the same species can look very, very different. Or even the same individual in different times, you can see how different it can look. So this would be a calcareous sponge, which the body is made of all these tubes that you can see that in the one picture they're very, very narrow. In the other one they are much more extended to almost become to almost make the sponge look like it's made of one mass. And the same the same specimen, same example sorry, same case of the same specimen looking different in different times in other species. It can be because of the shape that the sponge changes, or actually the color, or even the same specimen can look different in different areas because of some temporarily change of conditions of its physiology. So this is just to show a simple example that you can see of with your bare eyes of how sponges can be very difficult to identify. And this is now an example that is related to the skeleton of the sponges that I'm going to explain more in a minute. So I mentioned before that sponges have the skeletal elements that are called spicules. When I talk about sponges to kids, I simply say it's the sponge bones, is just to simplify. Really, so this is an example from Italy, as you can see from the map. So this species, crambe crambe, in the north of Italy shows only these two types of spicules, but in the Strait of Messina, where I come from, where in the water there is a much higher level of silica, this is the whole set of spicules that the sponges shows. So this is just to show how, again, sponges can be confusing to identify. But so to try to distinguish the different species, the main element to look at is their skeleton. So the skeleton is made of these spicules I mentioned now a few times already, and protein fibers made of a different type of collagen called spongin. So this on this slide I'm showing two different types of cells that produce the skeletal element in the sponges. So on the left-hand side on top, you have the sclerocyte, that is the type of sponge cell that produce the spicules, and on the other side you have a spongocyte, that is instead the cell producing these fibers of spongin that basically create the scaffold of the sponge skeleton. A limited number of sponges have their skeleton made only of these spongin scaffold, but the majority of them have these spicules organized within and around this spongin scaffold. So the spicules can be divided in macroscleres, which are the bigger spicules that create the main part of the skeleton of the sponges. You can see here they can have different shapes, and this is already a first way to distinguish different sponge species. But some sponge species may have only one type of these different types of spicule. Like an example is here. So to be able to distinguish different species, it's necessary to look not just at the shape, but also at the size of the spigule. If they are straight, if they are bended, if they have additional elements, like for instance, if you see on this diagram here, the shape C has this sort of circle midway through, or also looking at the tips of the spicules, so that they can vary largely. All these can be observed only at the microscope, and sometimes even very sophisticated microscopes are like the electron microscopes that I mentioned before, that allows to look at cell level, are necessary to be able to see all these very fine elements that help distinguish different spicules. But also sponges can have very small spicules that are called microscleres, that are used to furtherly support the skeleton. So the macroscleres mentioned before create the main part of the skeleton. These microscleres would be a sort of enforcement of it, and they are much more diversified, as you can see, so they greatly support the identification of sponges. So the diversification to distinguish the different species. Also, how the spicules, macro and microscleres, are arranged to create the skeleton. So within or around, as I mentioned before, the spongin fibers, is very, very important to be able to identify the different sponge species. However, to look at the spicules is not only enough. So what is called integrative taxonomy comes to help. So integrative taxonomy means looking at other elements that are diagnostic, so that they are useful to classify different species. In the case of sponges, one, for instance, is to look at the way they reproduce. So if they have direct development, or if they produce larvae, and how these larvae look like. That's an additional way to help distinguish the different species. Also, another way to help classifying the sponge species is to look at their different cell types. I mentioned before, there are several of them. One type is called the cells with inclusions, because these cells contain these more or less big vesicles that can have contain very different types of materials. Sometimes these famous bioactive compounds I mentioned before are stored in these inclusions, so in these vesicles inside these cells. And as you can see from the pictures here, each one of these is a different type of cells with inclusions, so they look very, very different to each other, and they also can help distinguish the different sponge species. But to be able to see this, we need again this very powerful electron microscope that I mentioned before. Otherwise, another way to be able to give a hopefully definite answer about which type of species we are dealing with is to identify them molecularly, so to extract some specific gene, sequence them, and try to see how they compare with others, so to be able in this way to name our target sponge species. So I'm gonna give you now some practical examples from my past of how all what I've tried to say with the last few slides. So how difficult can be to identify sponges. So all the species you see on this slide belong to a genus that is called Aliclona. That is one of the most famous to be difficult to classify. So all the species here have this particular type of spicule that is circled on the diagram there. So that already shows how looking at the spicules on their own, it's not enough to identify these species. And what I'm showing also is these are called phylogenetic trees. So these trees are constructed based on molecular data. So they show how the species analyzed relate to each other and other more or less related species. So this is first two examples that you see on the slide here, the picture on top of the two sponges. So please look on the right-hand side of the slide. The one on top is called Aliclona oculata. The one at the bottom is the one I showed you before, Aliclona similans. So they are both branching type of sponges. They can be found in the same environment. But if you look at the two smaller pictures on the other side of the slide, those two pictures represent are basically microscope pictures of the skeleton of these two species. So the one on top is again Aliclona cuculata, the one on the bottom is Aliclona similans. You see that they look quite different. And it's the Aliclona similans has much more spicules in its skeleton, and that's consistent with the texture of the sponge, which is very hard. So in this case, at least the skeleton arrangement helps to distinguish the two species, and the molecular data will do as well, because if you see the arrows, they indicate where these two species are on the phylogenetic tree, so they are actually quite distant from each other. This is now another example that of two other species, still Aliclona. The pretty pink sponge on top is called Aliclona mediterranea, and the purple at the bottom is Aliclona cinerea. If you look at the pictures representing their skeleton, they are actually not that different compared to each other. And if you look at the phylogenetic tree, they are not too far from each other either. So this is an example of more complex compared to the one seen before. But looking in terms of integrative taxonomy that, as I mentioned before, looks at all the possible elements that can help classify species. In the case of these two species, it's the geography that helps, because Aliclona mediterranea, as the name tells, can be found in the Mediterranean, not here. So that helps distinguish these two species. And now this last example shows these three sponges that from the top, you have the volcano sponge that Aliclona viscosa, then the one in the middle is called Aliclona indistincta, and the one at the bottom is called Aliclona sarai. So if you look at the pictures representing their skeleton, it's very similar, the type of mesh that their skeletons look like. They are very closely related, so they are very close on the phylogenetic tree. For Aliclona sarai, so the sponge in the picture at the bottom, it's at least it's the same case as we saw before. The geography helps. Sarai, again, it's a Mediterranean species. For viscosa and indistincta, so the other two sponges represented there, can be really, really challenging because, as you can see, they look very, very similar. And all the other elements we have seen so far don't help to distinguish them. So again, looking at the integrative taxonomy, other elements that we can look at to distinguish them are the cells with inclusions that I mentioned before. So if you look now, these 6 pictures, divide them in 2 groups of 3. So if you look at the first group on top, those pictures represent the inclusions that we can find, the cells with inclusions that we can find in Aliclona viscosa, Indistinta, and Sarai. So they look very similar to each other once again. So we can't use these elements either. So what really helps with these 3 species is to look at their microbial associations that we have seen. We talked before about these, the fact that sponges host different types of microbes. So in the 3 pictures at the bottom, you see all the arrows are pointing to bacteria. So it's something I would imagine the majority of you wouldn't be familiar with this type of images, so I don't go into many details, but it's just to, I hope you manage to see that what the arrows point at looks different in each picture, and that's because the first picture represents Aliclona Indistinta, the second one Viscosa, and the third one Sarai. So this is the only element that helps us distinguish these 3 species. So this shows how challenging could it be to identify these species. And here I just have this slide showing that another way to distinguish these different species or very closely related sponges is to look also at the chemical structure of the bioactive compounds they produce or can be extracted from them. So these are now some examples of resources that you can use to get more information about the sponge classification and the way to identify them. But now we get to the actual sponge project and how you can take part in it if you want. So you can collect, as I mentioned before, I'm actually realizing that it's already 10 to 9, so I'll try to shorten it. So if you want to send samples to us for them to be identified, as I explained before, we need from you a clear, one or more ideally, clear picture of the sponge, because that will help greatly the identification. For you to collect all the possible data describing the samples you have collected. And ideally, the samples should be stored in absolute ethanol, because that would allow the samples to be preserved in optimum condition for it to be analyzed with molecular techniques if necessary. And so when I said, now to go into details about what I just said there, when I said clear picture, what do I mean? So these essentially, all the pictures there with the happy face can help a lot us to identify the sponge. Definitely not something like the last picture there. That would be really, really hard to tell. So in terms of the details that you can give us to describe the sponge that can greatly help the identification is to describe the form of the sponge. Sponges can have very different forms, like can be branching, fan, cup, vase, tubular. They can be encrusting and massive as well, but encrusting encrusting and massive are all set of additional, they require a whole set of additional elements to be informative, because encrusting sponges can be very, very thin, or they can be thick. Massive can have different type of shapes. They can be lobes, they can be like balls, so they can be globular, they can have or not a stalk. So all these elements help a lot to distinguish different species. And then still about the form, sponges can have some specific characteristics that make them unique compared to others. So they may have pore sieves or projections or papillae. Some of these may really be the characteristic of a very small type, a very small number of species. So we may not be able to see it from the preserved sample or from the picture, even if the picture is not taken clearly. So all these elements are really, really important, as well as the description of the surface of the sponge in the natural environment. If it looked like it was spiky, or if you had a chance even to touch the sponge and see if it was spiky, if you could see the canals that I mentioned at the beginning of the presentation, these canals where the water runs basically from pore to oscules, or if you see any other evident features, like for instance the presence of a sort of mesh on the surface of the sponge that is characteristic of the Aliclona species that I mentioned before. So all these are very, very important details that help the identification, as well as the color. As written on the slide there, the color can be diagnostic to distinguish related sponges, because there are a few species with that all share these the presence of these pore sieves on their surface, but one species is red, one species is blue, one species is yellow. So the color can be really, really important, and the color tends to get lost once the sponge is preserved in ethanol. So it's very, very important to try to record the color immediately, so when it's fresh in the memory as well. Also because it can be characteristic of certain species. All the species you see in the pictures there is, in the picture at the bottom of the slide, there are no other species that have the same color. So that in these specific cases, the color is probably more important than many other elements to identify these species. Other features can be very useful. Sponges can produce slime, and as Tony here knows very, very well, because I really made his life a nightmare during my PhD, because I was looking for a particular species, Aliclona cinerea, that produces slime, and Aliclona cinerea looks very similar to other species, so at least another species which doesn't produce slime. So this is another very important element to record. If you see, you touch your sponge and you see that it produces slime, the surface can be very smooth, it can be rough, or it can be it can because of the spicules that can go through the surface of the sponge, can be pretty rough to touch it. So how it feels when you touch the surface is very important too. The consistency, as I mentioned before, Aliclona similans that is shown there is very, very hard. Aliclona oculata that may look similar if you're not familiar with them is actually not that hard. So even that element can help distinguish similarly looking species. The characteristics of the oscules, if they're big, small, if they have a particular shape, can help identify them. This is the picture that represents the volcano sponge that is called like that because of the shape of the oscules that look like small craters almost. The smell that can be, this is definitely personal, because sponges may not, they are filter feeders, as we said before. They filter water, they accumulate a lot of nasty smelling stuff, so they all smell a lot to you, but the smell can be actually diagnostic too. Believe it or not, there was a famous chemist in Naples who apparently was able to distinguish the sponges he was studying based on the smell and the habitat as well. I said I showed a few examples before when I was talking about the species from the Mediterranean, but even there are species in Ireland that they can be found in intertidal or and subtidal, or only intertidal, only subtidal. So the habitat where you actually have collected the sponges is very, very important as well. So this slide now is to show you, it's not a great slide now, I realize, because I don't have a reference there. But what I'm trying to say with this slide is that it's good to have, if you manage, if it's possible, it would be good to have a good chunk of sponge, because to prepare the sponge, the spicule preps, the skeleton preps, all these require sponge material, and it would be necessary, sorry, maybe to make more of this, more than once this this skeleton prep. So it's good to have some good amount of sponge there. Again, it would be good to meet in person to be able to show all these details with much more clarity. So again, in case you're interested in the sponge workshop. And another element of the sponge project of Search Island that is aimed at what I was saying at the beginning, to increase the records of these animals, is like a sub-project, let's say, that is called the Sponge Safari. So this doesn't require for you to collect any sample, but just when you go out in the ocean to, in this case, actually mostly underwater, because actually the majority of the species that I'm going to show can be found in subtidal environment. So to record the fact that you have seen these 6 species that I'm going to show you now, that when you see them in the way they are shown in these pictures, they cannot really be confused with other species. So the visual identification for this species is quite reliable. So the first one is the elephant hide sponge, which can be pretty big. It's a gray, pale gray sponge, pretty firm in consistency, can be recognized also by the fact that the oscules are normally collected in groups, and also at the surface, that's another element that I wanted to mention, it's pretty smooth. So this is one first sponge that can be easily identified with no need to go into all those details, all those techniques I mentioned before. Another one is the boring sponge, that is not called like that because of its attitude, obviously, but because of the boring sponge has 2 growth form. One is a massive growth form like the one depicted in the picture there. The other one is the boring sponge. It means that the sponge grows inside rocks, like limestone rocks, or actually even shells of other animals. And in those cases, what you see are only these sort of projections of the body, the sponge body, that are called papille. But this is one element that is really, really typical of the sponge is the color. It's this bright lemon yellow color that is cannot really be confused with anything else. The massive form, or actually, well, there will be another species, but looks pretty different. The massive growth form can get actually pretty big, and the oscules tend to be on the top of the sponge, and the papille that you can see better. If you look at the corner on the bottom right corner, you can see what I'm talking about. These sort of projections of the body are called papille, are characteristic of this species. Then we have the black tar sponge that is also there are no other black sponges. This sponge looks like a piece of rubber that had been stretched in between rocks. Again, the oscules are connected to each other, and it's it is like rubber. It's the consistency is quite tough, and again, another one that is pretty easy to identify is the crater sponge. It has these pore sieves that are very big. They almost touch each other. They are darker compared to the rest of the sponge, the body of the sponge, which has this lovely peachy color, and sometimes these pore sieves can be they can close up, so it might be more difficult to identify it. But when they are fully open, as you can see in the picture here, it's again difficult to distinguish the species with others. This one now here, it's the only example of calcareous sponge in our sponge safari. It's the purse sponge. These, from the picture here, it looks like they are massive. They are actually pretty small, even though they can get to almost 10 centimeters. They have this sort of light brown, even pale gray color, but they are actually they have a very characteristic shape. So they look almost like small sausages. One side is attached to the substratum, the other side where the oscule will be. So the oscule is surrounded by these it's almost skirt of long spicules, and that's you wouldn't really see this with other species. And finally, for our sponge safari, the last species is the chocolate finger sponge. That is called like that because of the color. It's another pretty unusual color. It's this dark brown reddish color. It's a branched type of sponge. The branches can be more or less longer, and they are pretty circular. The top is rounded. The surface of the sponge is pretty rough because, as I was mentioning before, the spicules go through the surface of it, and because of that, it can trap a lot of silt. So it may not look. So you may not be able to see the actual chocolatey color, but the shape and the fact that it's covered in silt could still help you identify it, even in environments like Albury here in Galway, where there is a lot of sediments. So again, these are more resources for you to be able. The resources I mentioned before are to learn more about sponges in general. These will be more to get familiar with the species in Ireland. And finally, I want to thank you all for your attention before asking if you have questions for me. I just want to tell you, if you want to learn more about this opportunity to have a sponge workshop with us, I have a QR code there that connects to a Google form that it's essentially an expression of interest. If you want to hear more about this opportunity, just fill this form so we get your email address and we can come back to you with more information. So I stop sharing the screen now.